PhD student at the French National Research Institute for Agriculture, Food and Environment. I'm going to present you through a case study how the biodegradation of antibiotic potentially confers an additional selective advantage for the implantation of antibiotic-resistant bacteria in the environment. Let's start with what we know. Antibiotics are considered as pollutants of emerging concern. Indeed, as they are widely used, poorly metabolized prior excretion by the body, and that their disposal processes lack efficiency, they are highly detected in many environments, with concentrations sometimes reaching nanograms per liter or even micrograms per kilogram. This pollution has plural ecotoxicological outcomes on environmental bacteria. First, by their biocidal action, antibiotics can alter the natural microbial communities and, by the way, disturb their function supporting ecosystem services. Second, more notorious outcome, pollution by antibiotics promotes the spread and the development of antibiotic-resistant bacteria. Consequently, the environment constitutes a reservoir of antibiotic resistances for pathogenic bacteria. Latest outcome discovered, and which interests us particularly, prolonged exposure of environmental bacteria to certain antibiotics has led to the emergence of catabolic functions. In other words, some bacteria could not only resist to antibiotics, but also use them as sources of nutrients. The risk-benefit study of this function, called antibiotrophy, remains to be done. On the one hand, it could be beneficial by increasing the self-purification potential of ecosystems and leading to the natural attenuation of their contamination by antibiotics. On the other hand, it could be of particular concern, as identified antibiotrophic bacteria are also antibiotic resistant. One may ask whether the antibiotrophy increases the dispersion potential of antibiotic-resistant bacteria in contaminated environments as the benefit of the antibiotic as dedicated nutrient source. To determine whether antibiotrophy facilitates the spread of antibiotic-resistant bacteria in contaminated environments, we used a study model based on sulfametazine. This bacteriostatic acts by inhibiting the bacterial dihydroapatite synthase and thus the nucleotide synthesis. Commonly used in veterinary care, it's one of the top 5 antibiotics most frequently found in the environment. Recently, some bacteria have demonstrated sulfametazine catabolic abilities. Among them, Microbacterium SPC448, which has been isolated in 2013 from experimental soils exposed for long to high concentration of the antibiotic. It is resistant to sulfametazine thanks to SUL1 gene and can also use it as source of carbon and nitrogen thanks to its 3 SAT genes. So, we had sulfametazine and Microbacterium as protagonists. We just had to find the scene. We selected the agricultural practice of soil amendment, which is known to strongly contribute to the spread of chemical pollutants and biological contaminants as antibiotic and antibiotic-resistant bacteria. So, why could it not also promote the dispersion of antibiotic bacteria? Her study was based on a microcosm experiment where 3 soils were or were not treated with a high concentration of sulfametazine, inoculated with the strain Microbacterium, and amended with manure. After 1 month of incubation, the establishment of the strain and its antibiotic gene were measured by real-time PCR, and the impacts of the treatment on soil bacterial communities were evaluated by 16S rDNA analyses. The degradation of sulfametazine was also measured during the months of incubation in additional microcosms, differing only by the addition of radiolabeled sulfametazine. With the establishment of the strain through the quantification of its GROL gene, which is represented on this graph. First, in red and gray, when it has not been inoculated, it was not detected, so the strain is not endemic of our studied soils. More surprisingly, in blue, when it was inoculated alone, it was neither quantified, indicating that the strain is a poor invader. However, in purple, the strain inoculated simultaneously to sulfametazine always succeeded in establishing. The minor amendment indicated by line type had no or few impact on it. By the way, the quantification of SAD-A gene had the same pattern, so it suggests that SAD-A was at least initially only hosted by Microbacterium. Two hypotheses could explain why sulfametazine is essential to Microbacterium establishment in the soil communities. Firstly, as the strain is an antibiotic, the antibiotic could serve as an exclusive trophic niche. Secondly, as the strain is also resistant to sulfametazine, it can benefit of its biocidal action, which can eliminate sensible trophic competitors. Mineralization kinetics of sulfametazine during the incubation represented on those graphs help us to explore our first hypothesis. In red, when the strain was not inoculated, the degradation were null or low, confirming that our soils have few natural degradation potential of sulfametazine. In purple, when the strain was established, the degradation rate varied depending on manure amendment modality. Indeed, with manure, sulfametazine mineralization was clearly increased. But when the strain was inoculated without manure, the natural degradation rate was not increased, except slightly in soil B, where it was anyway twofold lower than with manure. This result suggests that positive impact of sulfametazine on the strain establishment could partially rely on its nutritional advantage, especially in case of manure amendment, but in the condition where sulfametazine is not mineralized, the second hypothesis of biocidal advantage is clearly favored. To determine whether sulfametazine could have helped Microbacterium establishment through the reduction of trophic competition, we studied the response of the soil bacteria to the antibiotic treatment by performing 16S rDNA amplicon sequencing. Here are represented a phylogenetic tree of the bacteria whose abundance have been impacted negatively in red and positively in blue. The negative impact is summarized in the table. It has to be noticed that sulfametazine exerts a biocidal action on several bacteria, what may release trophic niches. Those may be used by Microbacterium to establish, instead of the antibiotic, what is in accordance with the absence of sulfametazine mineralization. Interestingly, the soil where the less bacteria were affected was the soil where mineralization of sulfametazine were observed, even in absence of manure. One could hypothesize that when the amount of trophic niches released is insufficient to ensure Microbacterium establishment, the strain used sulfametazine as nutrients, and the observed mineralization. However, the biocidal impact of sulfametazine is not diminished by manure amendment. So why does Microbacterium tend to degrade it in condition with manure? To find out, let's look at the impact of manure amendment on soil bacteria. One thing catches our attention, doesn't it? Yes, this blue square in soils A and B, which represent a cluster of 7 bacteria certainly originating from manure. We assume that they outcompeted Microbacterium to the occupation of niches released by sulfametazine. By the way, the strain is forced to degrade sulfametazine to ensure its establishment, explaining the observed mineralization. In soil C, we did not observe these bacteria brought by manure, so we assumed that the niches released by sulfametazine remains free from Microbacterium, which did not need to degrade sulfametazine. Let's summarize for those who fall asleep and for the drawing addicts. We had our soil communities and a bacteria with antibiotic degradation abilities which failed to invade it alone. But thanks to the biocidal action of the antibiotic which released some niches, the bacteria invasion succeeded. Well, the story is quite more complicated. Indeed, when the antibiotics were not enough efficient, the bacteria, dissatisfied, degraded. And when other invaders robbed its niche, the bacteria also degrade the antibiotic to establish. In conclusion, we show that the dispersion potential of Microbacterium is increased by sulfametazine for 2 reasons. As an antibiotic resistant, the strain benefits of its biocidal action which eliminates sensible trophic competitors. And as an antibiotic, the strain benefits of sulfametazine as nutrient source. If we come back to our first questioning about the risk-benefits of antibiotrophy, we can say that in terms of bioremediation, antibiotrophy interest is limited. Indeed, we showed that the antibiotic degradation was not systematic and had a low rate. Concerning the risk of increased dispersion, our results seems to indicate that the antibiotrophy allow dispersion of mycobacterium under conditions where an only resistant strain would not have succeeded. So, according to us, this emergent function may constitute a risk and should therefore be considered as such until further analysis confirm it or not. I would like to thank all the members of my teams, the members of the INER project, and you for your attention.